Now I am delighted to welcome Kate Maguire, Yuri Shevchenko, and Melina Mather, who will be joining myself and Gonzalo for our interactive roundtable discussion on the future of cancer research. So as before, please add your questions to the panelists using the Q&A box at the side of your screen. This is your chance to ask our speakers anything you want to know about the future of cancer research. And also don't forget to upvote your favorite questions. So while the audience think about their questions, I wanted to start us off with a question that hopefully puts a bit of a positive spin on the events that have affected us all over the last year, and ask each of you what you think the lasting positive effects of the SARS-CoV-2 pandemic will be on cancer research. Yuri, I'd like to start with you. What are your thoughts on this?
Well, what I've been thinking about, and maybe a little bit from a different end, is that now going through COVID, a lot of labs have been created that flourished specifically in the United States that brought in a lot of equipment to set up essentially just one PCR test, if you will. And what happens is there's a fair amount of automation and sort of power amassed at these centers. And now they will say, hey, if we could do this test, now we can test for anything. We can test for cancer. We can test for whatever. I come from the clinically regulated background. So I am a little concerned because I'm afraid that the next step will be appearance of many subpar tests on the market, rolled out by people who don't necessarily have prior, clinical knowledge, but saying, hey, well, we'll test for cancer. We have a big sequencer. We have a big robot. We're going to put together thousands of samples and just going to run them for cancer the same way. And so that a little bit concerns me. That may not be the sort of the optimistic subject so much, but I am naturally concerned about that.
And Yuri, how do you think we might get around that concern?
It's consumer education. Fortunately, most of these tests are prescribed by physicians, and hopefully bad tests won't be prescribed. So physicians are pretty educated. Now, as far as direct to consumer, that's pretty much what FDA should be doing. And if someone goes off in the lab, offering tests for whether your son is going to be a good soccer player or whether you have cancer or how soon you're going to have cancer, that kind of stuff, well, that should be quashed by FDA.
And Kate, what do you think the lasting positive effects of the SARS-CoV-2 pandemic will be on cancer research?
Well, I certainly agree. It will have a number of effects. I think one positive that I've seen is that with a lot of researchers needing to sort of transition to work remotely, I think we've gotten better and more comfortable with connecting with our colleagues that don't physically work in the same institution. It's always happened, but I think it's happening even more so at this point because of the pandemic. And I'm really excited to see, I think that's going to lead to broader collaborations and sort of more interconnectedness across the field, and maybe different groups not being quite so isolated. So I'm excited to see that happen.
And Melina, do you have anything you wanted to add to Kate or Yuri's comments there? Or do you have any thoughts of yourself?
Yeah, I'd like to add a few points. First off, I'll just say that the pandemic has really shown us the societal importance of investing in biomedical research. We've seen an unprecedented level of scientific collaboration and dissemination, which has led to research advances. And what's been really exciting is that it's culminated in the authorization of multiple vaccines against SARS-CoV-2 in less than 12 months after the first reported case of COVID-19. So this has been really kind of incredible and exciting to see such quick movement in the biopharma space. And it's really been transformative. And I think a lot of these learnings are also going to be applied to cancer research. Another one that I wanted to share was about just the changes that we'll likely see coming up in clinical trials as well. So the pandemic has been the biggest change to clinical trials that we've also seen in the last 30 to 40 years. And due to the pandemic, many medical facilities had to scale back operations last spring. And so I think this has really, encouraged us all to think more deeply, and especially clinical researchers to determine which procedures are absolutely necessary to do in person and which ones can be done remotely. And so I think this is going to have a huge impact this year and moving forward, and a positive impact where we might start to see increased access to clinical trials in the future for those who might not live near a research center and may not otherwise have a means to get there.
Fantastic. Gonzalo.
It's probably all been said now, but I think it's true. We have all access to much better and much more familiar working from home technology than we had before. I think this will help us do research across wider groups and places. I think it's very interesting to imagine what the possibilities are for these really molecularly guided and designed vaccines and antibody treatments that were designed and brought to market and approved and tested within a year. I think it's still a little bit fuzzy exactly how it will play out, but you could imagine that things like these mRNA vaccines will be very interesting for treating some cancers. You could have a vaccine that exposes you to cancer antigens and primes your body to protect itself. You could imagine that the ability to define these targeted antibodies and prove that they're safe and work in humans quickly will also be valuable in a variety of settings. And certainly for us at Regeneron, it was incredible to see how things that usually take many months or years to set up clinical trials across many sites were possible to do sometimes within weeks with really good engagement. And so, I think we're all hopeful that all these positive effects will come from, a super challenging time. But I think, probably some of them will last. So it'll be cool.
And there's a somewhat related question that has come in from the audience. And I think in the pandemic, one of the things that we've seen is this great collaboration between industry and academia. And one of the great examples of that is the Oxford AstraZeneca vaccine. And the question says, and it's directed at you, Kate, so I'll allow you to start, but from your point of view, how are the industry doing in terms of collaboration and helping move research from the lab to the clinic?
Well, I think, as you said, we've seen some great partnerships that have moved things forward much more quickly than usual. And it's my hope that that'll sort of be a model going forward, and that we'll be able to have better and maybe faster pipelines for translational medicine. But I don't have any specific examples of that right now.
I'd like to open it up to Yuri, Gonzalo, and Melina. What are your thoughts on this and translating research into the clinic? And some of the challenges that we might face there and how we might be able to get around that?
Well, so research setting kind of changes between the research setting and the sort of the clinical implementation. There's a huge gap, which is really I don't know how it just so happened. I'm responsible for the research part here at Semaphore, yet I'm talking about the regulatory, which is kind of the opposite end of it. I wish all the technology were out there and we could try it all, but the delay is inevitable, and implementation of any sort of researchy stuff usually runs against the ability to monetize. And that's what I'm seeing. If we could run the best technology that is exponential, if we could run it now for the patients, I think it would be very beneficial, but insurance company wouldn't think so, wouldn't pay for it, or it would be hard to draw some sort of clinical level conclusions, and so that's inevitably sort of me the gap between the core research and the clinical implementation of it.
And Gonzalo, a lot of your work has involved collaborations with academia and research. So what are your thoughts on this?
Okay, on collaboration or on translating from research to clinic, where should I say? These are very broad topics.
Sure. Well, let's start with translation.
So, honestly, when I think about translation, I'm probably like on the very far end of, nerdy, geeky kind of research questions, very, very basic. But I feel like, part of the issue as you move through that chain of taking a new discovery and getting it to the clinic is figuring out how you engage the different people that are involved in the process. I know that when we have a really cool genetic finding and we start talking with people who know, say, how to develop therapeutic molecules, which we do a lot here at, they will have different takes on what is a cool discovery that can be translated into a molecule. And so you need that back and forth, and then you start going into the details. Is this genetic finding in a transmembrane protein? Is it inside the membrane, outside, signaling domain? All those details end up making a difference on how you make that happen. Once you have that then you start thinking about what is the right place to try it in a clinic. What are the early outcomes where you try it? And again, you need engagement for people who know that side of things. It's in the end, it's almost impossible for any one of us to have the full picture of that process, and so it's, if you can figure out ways to get that back and forth dialogue going between people who know the very basic science, the translational science, and the clinic, that really helps. And when you only have one segment there, then you end up with these mismatches. Maybe someone's really great in the clinic, but they're very dated on the genetic technology, or someone's great at genetic technology, but they'll be dated on what's needed in the clinic or how to get it there. And I think then about collaboration, it is for a lot of these experiments that really need hundreds of thousands of samples and very large scales, it is the lifeblood of the process, right? Figuring out how you can get different people engaged and saying, this is a worthwhile thing to do together. It's you get much better quality answers with a large combined experiment than if we each do tiny experiments side by side.
And Melina, what about in the antibody discovery space?
About collaboration or?
About translation from research to clinic, and also about collaboration.
Yeah, I think I would agree with a lot of what Gonzalo just said about how it's very much a team effort with antibody discovery, and you need a lot of different functions to come together in order to bring it to the clinic. And these timelines, can be about a decade long, and so there's a lot that's needed in order to move things forward.
Okay, so I'm going to go to a question from our audience. So a lot of our speakers focus on areas that handle huge amounts of data. How is the advent of cloud-based and AI-based tools changing your field? Yuri, can I start with you?
Well, that's, the big data is what we're all about here. So as an example, well, again, back to how do you make sense of the data and what do you do with it, right? You can sequence an inherited cancer panel and say, hey, I know something about 50 genes here. And I'm just going to sequence 50 genes, and I'm going to send the clinical report out on these genes that I can clearly interpret. But at the same time, I can collect data on more than 50 genes and hope that someday, sooner or later, I'll have enough patients that I can draw some correlations, that I can do a discovery of some, modifiers and so on. Or I can take it even broader and say, hey, now I'm doing tests for hereditary cancer, but if they were to turn around and request another test, I already have the data. So boom, why don't we do the whole exome? Why don't we do the whole genome? And then there are ways to so how this data can be useful for the patients because we can review the data and come back with suggestions and so on. How this data could be useful for the pharma because they would want to somehow benefit from anonymized data sets. And I'm sure that Regeneron does that, a lot of that as well. And so I think that we've learned how to generate the data. Now is the next, sort of the time for the next level of expanded data sets beyond the minimum of what's needed. Thank you, Lumina, and for dropping the prices. And us digging deeper in informatics and AI, whatever you want to call it, related tools to make sense beyond of that data, beyond just the obvious areas of interest. And that's what we are hoping to do as a company.
Gonzalo, are there parallels that you can draw there with the work that you're doing at Regeneron?
Yeah, so I mean, the easy part of that question is the part about cloud computing. I mean, it's just completely essential to the way that we do work now. And I think, very few of us would go through the trouble of building a data center or a compute farm locally. It just makes more sense. You have much more flexibility doing it on the clouds. I mean, even imagine the little genotype by sequencing assays that we were describing. If you could run, several thousand samples in a week, and you could say, hey, let me request several thousand computers for a couple hours so I can process them all at once. You'd much rather do that than to either build a huge farm that you use for a couple hours a year, or then wait for fewer computers to get the work done. Machine learning is also really transformational, but it's about figuring out where to apply it. I think it's we sometimes have really big hopes for it in the sense that it'll be a tool that can solve almost anything, and it's not. You need to guide it. If you point it to good, interesting problems, it can be amazing. So certainly we've looked at data on let's mention tens of thousands of brains or whole body images with genetics, and it's because of machine learning that we can extract interesting features from that data, make sense of the images, align them. Then there's the big space about making predictions from machine learning. It's about, can you design good training experiments and have questions where you care about the answer? And I think we'll be seeing more and more of it, but yeah, definitely fun space to, if anybody is thinking about where they want to invest free time, it's a worthwhile space to explore, but it's very fuzzy still.
So Melina, I know that you just worked very closely with the Twist Biopharma team on a library that's been designed using AI. So can you talk a little bit about that?
Yeah, for sure. So we have a new library that we've come out with called the Twist AI Hypermutated Library that uses the power of AI to augment the design of a fully synthetic antibody library. And so what we're doing here is we have a neural network which mimics B-cell receptor recombination and hypermutation to produce antibodies with developability in mind. So, we're really looking at opportunities here where we can pull in some of that expertise to help design antibody libraries to better inform and to produce better diversities that our customers can select from for their antibody discovery needs.
And Kate, what about in the space where we're talking about, say, blood samples and samples that are maybe more difficult than standard samples to screen?
Yes, so we're actually moving towards doing a lot of our analyses in the cloud, and so getting back our sequencing data from blood biopsies, and then working in the Terra platform at the Broad Institute, which is basically runs on top of the Google Cloud infrastructure, and that's been really good, both in that it allows us to have put together standardized pipelines pretty easily, and also share them with other researchers, and to have all of our data in a central location that everyone can access, and to run things really quickly as well. So that's all been really helpful for our analyses. We haven't done as much with the machine learning, but we do have a project that we're working on now that's really exciting, and we found that we're finding a lot of really interesting patterns of classification of different cancers, and we're now working on sort of developing tools for our team to better interpret the output, because a lot of these machine learning tools are very black box, and so it's been really interesting to sort of try to untangle that and get actual biologic insight. So it's been really cool.
I'm looking at the time and I realize that we're going very close to the end of this session. But there is one more question here while I'm talking to you, Kate, that I think you would be able to help us answer here. And one of our audience members asks, so there's a huge interest in the adoption of the new canine exome globally. What are the processes that need to be completed for this to be released, and what does the timeline look like?
So we're really excited that there's so much interest. We have, as I talked about a little bit in my talk, we've done our pilot study and found that the metrics look really good, and so we didn't feel like we really needed to go back and readjust anything. So now there's just, I think, work taking place behind the scenes to get all the agreements that need to happen in place, and then hopefully as soon as that happens, then people will be able to order it. And I just don't have a great sense of how long these sorts of things tend to take, but I know that it's a high priority for everybody, so hopefully it won't be too long.
Well, thank you. So I'm afraid that we have run out of time, but I would like to thank Gonzalo, Kate, Yuri, and Melina for their insights and also your contributions to this really interesting discussion.